All right, we're going to get started. Thank you, everybody. I feel like I'm back in school where everybody sits in the back of the room. Nobody wants to sit at the beginning because we're going to call on you if you're in the back of the room. Just kidding. So welcome. I'm Lynn Schnapp. For those of you who don't know me, the chair of the Department of Medicine, and we are absolutely delighted to bring back Research Day. Although, as I say, every day is Research Day. And I want to thank Dr. Oziama Okanwa, who has, and the research committee, who has helped design Research Day. So without further ado, we've got a very exciting agenda. I hope you'll stick around. We've got posters, we've got additional sessions, we've got research awards. And so with that, I am going to introduce first Oziama to come up. Oziama is an associate professor in the Division of Geriatrics, and he is going to be introducing our special guest speaker.
All right, thanks so much, Dr. Schnapp, for that kind introduction. And now it's my distinct pleasure and honor to welcome you all to Research Day and then to introduce our grand round speaker. Dr. Sid O'Bryant is the Dr. Joe and Peggy Schuler Endowed Chair in Pharmacology and Neuroscience, and also the Executive Director of the Institute of Translational Research at the University of North Texas Health Science Center. Dr. O'Bryant is the principal investigator of the Health and Aging Brain Study Health Disparities, short for HABS-HD, which is the most comprehensive study of Alzheimer's disease among the three largest racial ethnic groups in the US: African Americans, Mexican Americans, and non-Hispanic whites. The goal of the HABS-HD program is to understand the life course factors, including biological, sociocultural, environmental, behavioral, that impact risk for Alzheimer's disease in late life. This work will ultimately lead to population-specific precision medicine approaches to treating and preventing Alzheimer's disease. That is, treating your Alzheimer's disease. In addition to being a global leader in Mexican-American cognitive aging, Dr. O'Bryant is a global expert in precision medicine approaches to novel diagnostic and therapeutic approaches to Alzheimer's disease, Parkinson's, dementia with Lewy bodies, and Down syndrome. In addition, I have the honor of calling him friend. So welcome, Dr. Sid O'Bryant.
All right, can you hear? Is it on? All right, making sure it's actually on. Timer. So first, thank you. It's really an honor to be here. Get to see friends that I've known for a very long time. Oziama and I've been friends for many, many years. Amy, Sterling. It's nice to be here. So I'm going to talk to you about Alzheimer's across diverse populations. And I named it the way I did to be a bit provocative, because my opening statement is going to be something that some people don't like. But it's that I believe that our conceptualization of Alzheimer's is wrong. I think that it is too narrow and it is too myopic. And 90% to 95% of all individuals enrolled in Alzheimer's research have been non-Hispanic whites. And that ignores the scientific literature from oncology, endocrinology, heart disease that show that racial and ethnic factors are very important, not only for the presentation of numerous diseases, but even therapeutics. In the oncology space, there's numerous drugs that actually have racial and ethnic designations in the indications for the therapeutics themselves. And so in brain health, we've largely ignored this for years. But however, in the last, I don't know, I guess 10 years, there have been huge advancements in this line of work. And I've been having the honor of doing this for a very long time. Those are disclosures for et cetera. This is the most important slide that I will show today. And this is actually only my team. This isn't the actual full collaborative list. This is the Institute for Translational Research with some of the collaborators listed on the right. But I tell everybody all the time, I don't actually do anything. As a team, we do a ton. And this is, it's an honor and privilege to be able to lead this amazing group of people. Okay, so let's jump in. Why is this important? So many of you will know, many of you will not. Alzheimer's disease disproportionately impacts communities across the globe, not just in this country, but in this country, Alzheimer's is more prevalent, and dementia in general, Alzheimer's and Alzheimer's-related dementias are more prevalent among underserved communities, particularly African Americans and Hispanics. 90 to 95% of the research that built some of our key guiding frameworks have been based on non-Hispanic white individuals, and less than 5% of individuals enrolled in clinical trials have been from diverse communities. However, that ATN framework that now guides the field hasn't been examined. So ATN, for those of you who are not Alzheimer's specialists, is amyloid, tau, neurodegeneration. And so amyloid is just plaques, tau is just tangles, and neurodegeneration is brain shrinkage. And so those are the three considered fundamental pathological mechanisms for the disease. And that framework has not been studied, as I've said, so that's one of the things we're pushing very strongly. So why is this becoming more and more important? Well, the demographic population of this country is changing, and it's been changing for many years, and it will continue to change over the next several decades, where the proportion of the population that is non-Hispanic white is now going down under 50%, whereas the proportion of Hispanics, African Americans, and other racial and ethnic groups are growing in this country. And I don't think our census data is actually keeping up with some of the changes, especially in Texas. So why is Texas kind of an interesting place for this work? Well, Dallas-Fort Worth, where I live, is one of the largest metropolitan areas in this country. There's over 7 million people. However, what a lot of people don't realize is that DFW currently is reflective of the racial and ethnic demographic characteristics of where the United States will be by 2030, 2040. So Texas DFW is already there. So it makes it a really kind of an ideal location to work with diverse communities, which is one of the reasons why I've moved to that community 10 years ago. So what is HABS-HD? Some of the original publications you will still see out there called ABLE, Health and Aging Brain Among Latino Elders. The study's always been the Health and Aging Brain study. However, we do a community-based approach to reaching participants. So we have targeted outreach for the communities themselves. And so ABLE was and is the name of the targeted recruitment to the Hispanic community. We're now kind of shifting some of the logos, et cetera, to try and be a little more consistent. We've already enrolled over 1,000 non-Hispanic whites and 1,000 Mexican Americans and over 700 African Americans. The original cohort was built only studying Mexican Americans and non-Hispanic whites. We were able to secure funding to add the African American component. So now that's where we are. With the recent renewal of the project, we're expanding down to 30 and above to study the life course natures and adding 500 per group. So 1,500 Hispanic, 1,500 non-Hispanic white, and 1,500 African-American. One of the things we also recently did, the reason we started studying Mexican-American is because Mexican-Americans are 65% of the US Hispanic population and 80% of the Hispanic population in Texas. However, that means we were not representing 20% of our own community. So we have now changed the enrollment. We're now enrolling Hispanics of any origin, country of origin, to be better reflective of our own community. I start off with describing the study because we're going to talk a lot about data from the study. Many of the data, and I say this up front, this is an open access data study. So any of you can request access to any of the data at any time. The portal is already there. We've already given out over 150 databases in 7 countries since January. We do functional exams, clinical labs. We examine sociocultural factors, cognitive assessments. Biorepository now is closer to 1 million aliquots. And this is a full systems biology study. So we're capturing MRI, amyloid PET, tau PET. Then we do, or we will be with the expansion, whole genome sequencing, GWAS, methylation, untargeted and targeted proteomics, untargeted targeted metabolomics, transcriptomics, and exosome. So that's a lot to get out. So it is the most comprehensive study of the biology of brain aging among diverse communities ever conducted, but it's all done within a health disparities framework. And so Amy's group is doing the ADI, not only current residents, but now we're going to be doing longitudinal, so historical residents, both in the United States and in Mexico. Okay, so this is how the study comes together. Ozayama leads the development core. Amy's part of it. Brad is also part of the study. And here are the co-Is, et cetera. Okay, this is a bit of a dated slide. It's now over, actually, it's over 800 African Americans. But what you see is, there's a different prevalence rates of MCI, which was also Jen just published from the Health and Retirement Study. The average age is younger because our cohort, the entry age isn't 60 or 65, it's 50. Now we're actually enrolling 30 and above. And so the age range will continue to, the average age will continue to go down. Now, what really got us moving into this was, so back in 2013, part of the Texas Alzheimer's Research Consortium, and I published an article where we found that Mexican Americans' average age of being clinically classified with mild cognitive impairment was substantially younger than that of non-Hispanic whites. And then that was subsequently published by other investigators that were finding similar things, increased risk for MCI as well as at younger ages. At that point, we didn't have imaging, and I'm a neuropsychologist by training. And so the first thing we started thinking about is, were our tests biased? Were we over pathologizing due to inappropriate norms, et cetera? We built our own local norms, however, so that all of our tests are actually normed for our population because we have a large enough sample size to do so. So, and anyone involved in our consensus diagnoses processes have extensive experience looking at neuropsych files and files from diverse communities. So we didn't think that was the issue, but we still keep an eye on that. So the first of the biological markers that we decided to look at was ApoE4. So ApoE4 is the single strongest genetic risk factor for late onset Alzheimer's. I'm sure most of you know that. However, back in the 90s, there was a publication looking at ApoE4 across the globe and showed very strong differences across communities globally. And what we found was lower rates of ApoE4 positivity among Mexican Americans, and Hector Gonzalez subsequently validated that in the Sol Inca study among their cohort. So what we found, we start digging deeper, and what we find is, okay, increased risk for cognitive loss at younger ages, but in the absence of the primary genetic risk factor. We started looking at medical comorbidities, et cetera, and you see drastically different prevalence rates. And so this is unpublished data that one of our students is working on. We've done GWAS now on around 800 Mexican American and 800 non-Hispanic whites. And we just pulled some of the top genetic SNPs from Kunkel et al. And basically, all SNPs vary. It really doesn't matter which one you look at. And so we're saying, okay, so we see different prevalence rates, different risk factors, and even the different genetic SNPs. So that's then what led us to say, okay, the ATN framework came out in 2018. And the whole point of that framework was to build a research classification for advancing novel therapeutic trials. It's not used clinically because most people don't have access to amyloid scans, lumbar punctures, or whatever, at least not in this country. So our first place to start was amyloid. And the reason amyloid is because with the recent framework, Alzheimer's disease from a research standpoint means amyloid. You cannot be classified on the Alzheimer's spectrum biologically in the absence of amyloid pathology. Now that it poses some interesting questions we're going to get to later on. And so that's where we started to look. And what we found was, as you can see, amyloid positivity rates across diverse communities substantially lower. And so in HABS HD, we now, I think as of this week, we will have surpassed 2,000 amyloid PET scans in the cohort. We have over 1,000 tau scans at this point. So there's like 6,000 MRIs. So there's a ton of data. This is some of the earlier cuts of the data. And so we find this lower prevalence rate of amyloid positivity across racial and ethnic groups, both Hispanic and African American. We wanted to, all right, what about other studies? So we just wrapped up the first ever primary care study of a blood test, basically, for primary care. Nothing's been published on that one yet, but the amyloid rates, we found the same thing. Robert Rissman and I have an R01 looking in the A4 data, and we found the same thing there. So it's this interesting position to be in when you start saying, okay, Alzheimer's disease is amyloid. Hispanics are at increased risk for Alzheimer's disease. African Americans are at increased risk for Alzheimer's disease. But now what we find is lower rates of ApoE4 positivity and lower rates of amyloid positivity. So what exactly are we seeing? However, one of the questions that we had, and that I was very keen on asking is, okay, so, but in the presence of, say, ApoE4, do some of the thought kind of, right now, the general notion is ApoE4 is somehow strongly linked to amyloid. Well, what we found is that in the presence of ApoE4, it is still strongly related to amyloid burden among Mexican Americans, but even then the link is weaker. It's not as strongly related to amyloid as it is among non-Hispanic whites. So, okay, what is related to amyloid burden? And so we did a pilot study and found that metabolic markers were more strongly related to amyloid among non-Hispanic whites. But then one of the things that kind of has been a little bit of a dilemma for the field is amyloid is pathological marker for Alzheimer's, but amyloid doesn't track very well with cognition. Presence of amyloid doesn't mean you have cognitive impairment. Presence of amyloid doesn't really predict cognitive loss very well. So does it predict cognitive outcomes? And what we found in this cohort, this cut of the cohort, still 210 African American, 100 Mexican American, there were about 200 non-Hispanic white. Amyloid was related to cognition, but specifically among African Americans. Amyloid, as has been published previously, wasn't really related to cognition among non-Hispanic whites, but it was a little bit among Mexican Americans. And so this picture starts to get even more muddy, so to speak. So what we have is increased risk for dementia at younger ages in the absence of ApoE4, lower rates of amyloid, but when amyloid or ApoE4 are present, they are associated with pathological outcomes among diverse communities. So, all right, what about other markers? So this study, what we did was looked at, I mean, there's so many different ways to look at neurodegeneration on an MRI. So what we took was Cliff Jack's just meta ROI. It's basically an Alzheimer's cortical signature. And what we found was in positivity, look at the age difference. So that in positivity is happening, co-occurring with that cognitive loss. So we see in neurodegeneration positivity at younger ages among our Mexican American participants. And I'm describing most data among Mexican Americans, non-Hispanic whites, because the African American cohort was only recruited more recently, so not all that data is yet available. But we are looking at this across groups. And so we went back and looked, and neurodegeneration is related to the cognitive outcomes that you would think it's related. It's associated with poor memory, et cetera. So this is a project by junior investigator Ali Clark in UT Austin. And so one of the big initiatives of our cohort and our study is, okay, so we're finding these differential prevalence rates of these core pathological markers, but we're finding different outcomes and different relations. It doesn't fall and fit with the normal conceptualized pattern. So if you think of the Jack curves of, some of the conceptualizations would be amyloid, then tau, then neurodegeneration, then cognitive loss. We're saying cognitive loss actually happens before potentially amyloid building, but then neurodegeneration is probably happening before accumulation of amyloid as well, at least at this point. So what are the other factors? What's driving this, especially neurodegeneration? And so what Ali did was pulled, and her interest is looking at some of these, how are these cardiovascular and metabolic factors associated with cognitive loss? And what she found was fatty acid binding protein was associated with both white matter disease and neurodegeneration. And the link was strongest among Hispanics. It wasn't really as significant among non-Hispanic whites. And this is consistent with some other data where we've seen that glucagon, for example, was related to amyloid burden among Hispanics. And other data now that is being looked at is showing that other markers of insulin, for example, Meredith Brasky's presenting some data soon on this, is insulin also seems to be related to white matter disease and neurodegeneration among Hispanics, but not non-Hispanic whites. So what we're seeing is some sort of metabolic something going on that may be driving cognitive loss. But it's important for those of you who are clinicians to remember that most of these people are still presenting memory loss, language issues, maybe some executive functioning problems. And so what we're seeing looks like an Alzheimer's pattern, but the underlying factors associated with these outcomes seems to vary. And pardon me, this is where, so we started looking at tau PET. And so we have now a little bit over 1,000 tau PET scans, but in a study like this, collecting data versus getting the data out, there's a gap. But what we found very early on, and actually Amy and I were talking about this last night, is I had to email Bruce Miller about this, because early data we were seeing looked like tau positivity was higher among Mexican Americans. Well, the general notion is it's amyloid then tau. And what we found in this study, this was a poster presentation, was that tau was strongly associated with cognitive outcomes, specifically on Mexican Americans. There was really not much going on among non-Hispanic whites, some significance among African Americans. And so let me pause for a second, because these data are very conflicting with the general notion today of the field. So what we're seeing is differential prevalence rates, younger ages of onset, but then once you dig into the biology, lower rates of ApoE4, lower rates of amyloid pathology, and now what we're seeing is even amyloid and tau are differentially associated with cognitive outcomes. And so now that we have disease-modifying therapies on market, hitting the market, a tau agent's probably going to be hitting the market soon, these findings become even more important. And so this was an interesting slide to pull together, and I think, Amy, this is the one you saw last week at the Alzheimer's Centers meeting. And so we haven't published this yet, but sample sizes, you can see, are significant relative to what most studies, have done. And it doesn't matter really which of the ATN markers you look at, positivity rates vary by race and ethnicity. And one of the things we're trying to now start digging into is our early data suggests that the sequence and timing of the markers may vary as well. And there's some very interesting research that's been coming out for several years now on the general notion that, positivity is really not the important thing. It's where is the amyloid, where is the tau, or where is the neurodegeneration? And that's data that we're looking into now to see if does the location of the presence actually matter differentially. I can tell you that depending on where and how you set the cut scores, the positivity rates across different ROIs varies across groups. And so what we don't know is, are the cut scores appropriate across groups as well? So one of the things that we've started looking into a little bit, because when many of you do trials, and in the Alzheimer's space, it's oftentimes it's what are the ADLs, and ADLs become significant outcomes, and then it's also just kind of the motor components and what's going on during aging. And so this was a very interesting project where what we wanted to look at is just general motor function, physical functions, just timed up and go in the short portable physical battery. And what we found is that these plasma markers of Alzheimer's pathology were differentially related to physical outcomes, as well as we've already shown cognitive outcomes. And so what you see here, and this one was general neurodegeneration as measured by plasma NFL, was only associated with functional outcomes among Mexican Americans. That's the marker I think we all figured would be associated with functional outcomes across everyone, but that's just not the case. Where I've spent a ton of my life also is in the blood-based biomarkers. And one of the things that I've personally, one of my scientific areas, is to try and identify screening tools for primary care docs. Most patients with Alzheimer's disease never see a specialist in this country. It's only about 10% of Alzheimer's patients actually ever see a specialist. Most patients are seen, treated, monitored by their primary care physicians. There's also a ton of literature showing that patients or individuals from underserved communities rarely, if ever, go to specialty clinics. And so in our Hispanic cohort and our African-American, what we see is most of these individuals, they never go to a specialist. And so what we've been trying to work on is, can you create a blood-based system, a blood for screening in primary care to determine who should be referred over? access to these PET scans and MRIs is limited. And one of the other things that I think, if for those of you who end up working with underserved communities, is our own assumptions and biases can really get in our way. When we started doing MRIs, the vast majority of individuals that were coming through our imaging center, not only had they never had an MRI, they didn't even know anyone who had an MRI. And so when you get into, 45 minutes into an MRI scan and that DTI sequence hits at the last 10 minutes, and it sounds like a jackhammer going off in your ears, my ears have been ringing, and now I have ear damage, and it's like we hear these weird things, but it's because of lack of access and lack of awareness. Same thing with PET scans. It's hard still trying to wrap my head around it, but when we've been talking to underserved communities, and actually there's an article about to come out on this, when you start discussing PET scans and you say, well, we're going to look at these buildups of these proteins in your brain, it gets misunderstood as an autopsy. And when we first got that feedback, it's like, oh my goodness, wait a minute. They think we're asking them to donate their brain because we've said we're going to look at what's in your brain. And so we had to take steps back of just how do we phrase the research itself. And I remember Miguel, one of the people who used to work for me, was when he first started running the pilot study for this, he's like, Dr. O'Brien, I don't really like the word radio tracer. Like, well, okay, no one likes the word radio tracer. And so how do you articulate these really advanced medical procedures in a manner that someone with 0 years of formal education understands in an acceptable manner that they can provide consent to come into a research study? And so it gets very complicated. And so one of the reasons we've been in the blood-based biomarker space is so we can facilitate access to these advanced diagnostics. And we've been working on blood tests for Alzheimer's disease for longer than I care to admit. And in this study, what we found was fascinating, because in our pilot work, we were finding an AUC of always 90, 95%, very good diagnostic accuracy. But then we said, okay, how does the proteomic profile look across groups? And in our early work in 2014, we found that you could take the same profile of, let's just say, 30 markers, and it would still be accurate among Hispanics, but the relative importance of the markers was completely different. And so in this study, we applied this to several hundreds of individuals from both Mexican American, non-Hispanic white cohort. And what we found is the accuracy, as you can see, the AUC of 91%, this is for MCI, 94% among non-Hispanic whites. So the accuracy is really good. However, the proteomic profiles themselves are drastically different. And so what you'll see, I know it's hard to see it, but for example, amyloid, tau, et cetera, A beta, et cetera, only rank in the top half of proteins for non-Hispanic whites. They do not rank in the top among Mexican Americans. And what you also see, if you look at the top half of markers for Mexican Americans, you see GLP-1, you see glucagon, you see FABP3, insulin is in there. Among non-Hispanic whites, you see insulin, but then the metabolic factors kind of drop down. So you see a more metabolic profile of MCI among Mexican Americans, vascular, inflammatory among non-Hispanic whites. But when you get to dementia, it changes. So GLP-1 is now the #1 predictor among non-Hispanic whites for dementia, glucagon #3, whereas you see now a lot of the inflammatory markers increase among Mexican Americans. And so it's not only do we see different proteomic profiles of disease itself, but the profiles change over the course of time and the disease progression. And there's some research out there looking at like neurotropic factors and how they change. You may see things dip and then go up and then fall off a cliff. It's same with inflammatory markers. Hasn't been studied as much in neurodegenerative space as obviously it needs to be. Plasma NFL has, many of you know, it's been like taken off. Everybody is been interested in NFL in the neurodegenerative space for several years now, and it seems to be really significantly associated with a ton of outcomes. And plasma neurofilament light, thank you. Neurofilament light chain. There's still neurofilament heavy chain, which haven't really looked at a lot in the literature, but neurofilament light, interestingly enough, it's kind of a newer marker for the neurodegenerative space. It is not a newer marker for multiple sclerosis, for peripheral neuropathy, and other diseases. As a matter of fact, people have been looking at this and potentially tracking progression in multiple sclerosis clinical trials for some time. And folks working in peripheral neuropathy have been using plasma NFL as an indicator of motor problems for a while as well. So what we did was said, okay, let's look at it. You know, is it strongly related to cognition? And it was, but it's also related to MRI outcomes. So it's considered to be a peripheral marker of neurodegeneration, but it is not a peripheral marker of central neurodegeneration only. It's a much more complicated marker. And for those of you who do cognitive testing, the best example I can give you is plasma neurofilament light is like Trails B. It's sensitive to everything and specific to nothing. So you know that it's always related to bad outcomes, but you don't really know what it means. But what we found is even plasma NFL levels vary. So even in the blood-based biomarker space, these things vary. And I didn't keep the slide. One of the things that, because of this, what we found is these differential markers and how they come together, different medical comorbidities. We also looked at how medical comorbidities are associated with these plasma markers. So, you know, up until recently, these plasma markers were, you know, when will we be able to have access to them? When will they be facilitating access to trials and care? But these markers are now clinically available. Plasma NFL is clinically available. Several of your plasma amyloids are clinically available. And even some of the p-taus are soon to be clinically available. And what we found, and Michelle Mielke was the first to actually, I don't take credit for this, she published this out of Mayo data and found that you know, diabetes, hypertension significantly alter plasma biomarkers. So she and I were talking, and she told me to look at it in our cohort because, you know, these medical comorbidities are differentially prevalent. And what we found is consistent with what she found is that these medical comorbidities are very strongly associated with these plasma outcomes, especially mild kidney issues. And so EGFR levels not in clinically pathological levels very strongly relate to these markers. And so how do we interpret that and how do we assimilate that into clinical practice is completely unknown. There are no guidelines at this point of really how to take and use these markers. There's a ton of other data that I could go through, and I know I'm probably beating you up with more data than you want, but I feel like I still have to convince people that this is important. And so what you see, what we've seen is it almost doesn't matter where you look into these different biological mechanisms. You can look at amyloid, tau, neurodegeneration, inflammation, metabolic dysfunction, vascular dysfunction. Doesn't matter where you look. They're all still important. Just like among non-Hispanic whites, they're still important, but the prevalence of them varies. And so the way I see this is the relative weights of these different pathological systems appear to vary. Why they vary, though, we don't know. Partially, I'm assuming due to medical comorbidities, but even before I jump into this, even thinking back to some of the earlier data I showed around ApoE4, well, there's recent data showing that ApoE4, the impact of this as a risk factor for Alzheimer's disease is moderated or mediated by genetic ancestry. So ApoE4 in the presence of African ancestry is not a risk factor. ApoE4 in the presence of European ancestry is a risk factor. And so among Hispanics, we just don't know yet. So it's more complicated. But then there's the sociocultural components. And so this is another thing that we feel, and I feel very strongly about. I don't believe one can understand the biology of any disease in the absence of the environment within which someone lives. And that environment isn't just, when I say environment, I don't just mean environmental exposures in the classic sense of, you know, I grew up in South Louisiana. There were 7 Superfund sites in Louisiana when I lived there. I don't just mean that. I also mean what are, you know, the racism, perceived racism, cultural identity, et cetera. All of these things are important for understanding brain aging. And so this is one, this article, I actually just talked to the group just the other day. This is, so you'll be seeing a manuscript soon, a draft. And so we worked with Amy's group and we created the current ADI. This isn't historical, just current. And one of the things that I wanted to do just, it's kind of a Captain Obvious sort of thing, but when you see the data, it's one of those that kind of shocks you. So these are the quintiles of ADIs. And so by Mexican American, non-Hispanic white. So you have greatest area of deprivation, least area of deprivation. And so what you see is a mirror image like that. And so, and the percentage is almost completely flip-flop. And so you see 40% of non-Hispanic whites live in the area of least deprivation, 33% of Mexican Americans in the highest area of deprivation, 3%, 8%. And so what we're seeing is there's just this flip-flop of where people live, which is not surprising, but to look at the graph when I saw it still is a bit shocking. But even more importantly is look at the table next to it. And so I can't even point anymore. Oh, I think I broke your, oh, there it is. I break things, sorry. But if you look at matching within ADIs, Mexican Americans have lower levels of education and lower annual income. So even living in the same geographic region does not equate to same access to resources. However, as Amy has shown numerous times, ADIs are still related to poor cognitive outcomes. Really doesn't matter what test that you look at. Now, there's a recent, in our study, what we just found is then we said, all right, well, let's look at cognition within ADIs. And when you look at cognition within ADIs, we didn't see the racial and ethnic differences. However, what I'm betting is we will see different progression rates. And so, and that's one of the things we've talked about a lot is the dosage and timing of things that we're going to be digging into. So I'm going to wrap up and then talk a little bit about data access because I want all of you to take the data. So what are kind of the take home points of what I have to say to you today? Well, like I started with, I always believe and begin with the end in mind. What did I say at the beginning? Our conceptualization of Alzheimer's disease is wrong. It's not that deep. I'm not saying it's wrong with regards to the importance of these pathological markers. These markers are still very important. What we have to do, though, is take a step back and broaden our conceptualization of the disease. Because one of the clinical dilemmas that people are running into now, and I was sharing this today, I believe, you know, I have a friend who referred someone into the study. They hadn't received their feedback. They were texting me what's going on. And I'm like, oh, it's, you know, it's going. And I grabbed the file, I'm looking at it. 84-year-old, non-Hispanic white, 14 years of education, female, poor encoding, rapid decay, poor language, expressive language, and some mild executive issues, macrovascular disease, amyloid negative. Just looked like an amyloid patient, looked like an Alzheimer's patient, and diagnosed clinically with Alzheimer's disease. She's going to get a feedback letter from us that says dementia, not Alzheimer's disease. And so now we're working with our own community and the physicians to say, okay, well, what now? She's already on Aricept. What do you do? Well, fortunately, there's data to suggest Aricept's actually okay for people who have vascular disease, but this isn't your classic vascular dementia. She has chronic microvascular disease, but no strokes. Well, we're seeing that people who clinically that we see look like Alzheimer's disease among non-Hispanic whites around 50 to 60% amyloid positivity, but among African Americans and Hispanics, it's under 50%. So that means less than 50% of people walking through with a dementia diagnosis that look like Alzheimer's would be correctly classified as Alzheimer's disease. And so given the state of affairs with drugs coming out, et cetera, this poses some interesting problems for us as a field that we have to figure out. And that said, we're working with our own local physicians and clinicians of how do we present that back to the patient? They're not my patient, this is their patient. And now so it's complicated. The other thing though, is these biomarkers are clinically available. You can get access to these blood tests now. You can get access to these scans. I will tell you that, and I haven't published this, but the agreement between clinical read of amyloid positivity and SUVR cut-based positivity, the agreement among non-Hispanic whites is 70% in ours, which is kind of close. It was less than 10% among our underserved communities. And so it's like, all right, how is this and what's going on here? So we cannot advance true precision medicine without inclusion of diverse populations. The complexities we're seeing now, in my opinion, are the keys to beating these issues in the first place, because we can target these individual pathologies that have different prevalences. And so we need to know who to give an Alzheimer's drug, an amyloid drug to. We still don't know, just because someone has amyloid doesn't mean that they need an amyloid drug, because we don't know who has amyloid-driven cognitive loss. And so these are things that we need to really spend more time on. And without looking among diverse communities, we're failing our own community, in my opinion. And so, the way the study is designed is we have overlap with tons of other projects, including your own. The data, this is open access. So all you have to do is go to the website, create an account, submit a proposal, it'll get approved, and the system automatically generates a database for you and comes back. With the new grant, all data will be moving over to Lonnie soon, but we still access to everything. Raw scans are completely available if you want to reprocess them. Any derived data you create must go back into the study itself and access to the scientific community. We've already supported, I think, 4 ancillary RO1s, and I think we're currently supporting 6 training grants, K grants, American Heart Association, Alzheimer's Association, you name it. And so I don't view this as my study. I view this as our study. It belongs to the scientific community. It belongs to my community in Fort Worth. And so I can't help patients if you don't take the data and do things with it. So that's my shameless plug of please use our data. So what's in the database currently? Visit 1, that's what was in the database at that point for visit 1 and visit 2. However, we've now did an updated data release that was in July. The new database release is coming out, and this is what should be in the database at that point. There will be some visit 3s even, but then there should the PET scans will be in greater and greater numbers. At the moment, we do now have almost 1,200 participants across diverse communities with tau, amyloid, and MRI. And so tau is the last component of the study. It's actually an independent IRB because of the FDA and other things. So if you want to look at all 3, if you start with tau and go backwards, you can find anyone in the database that has everything you want. I'm going to stop there. Take questions. Bless you.
Thank you, Dr. O'Brien. That was a wonderful presentation. And I already see hands up in the audience. So I'm going to start from over here, Dr. Johnson.
Yeah.
There we go.
Really terrific talk and very provocative. First, I would just want to thank you for this, and also for your leadership in the plasma biomarker space, where you were flying the flag when no one thought it was possible, and now it's a reality, and I want to thank you for that. My question is about the idea of multi-pathology dementia. We know TDP-43 is a thing, and TDP-43 or alpha-synuclein is often present in people who have Alzheimer's neuropathologic change, and they could be present in and of themselves and cause multi-domain dementia. What do you think is going on there? And also adding in vascular disease, that's another pathology. How can is there a way to stick to the ATN and just broaden the idea that we have multiple pathologies in addition to ATN pathology?
That's exactly what we've proposed. As a matter of fact, in the U19, we officially proposed an ATNVMI, vascular metabolic inflammatory. And the only reason, if you go to my office, you'll also see alpha-synuclein and TDP-43. The only reason we didn't include those is getting at them at this point. So the plasma markers of alpha-synuclein are just a mess. TDP-43, we're still trying to figure out how to get at good ways at doing that. My hope is to do DAT scans. I'd love to do DAT scans on people, because if you look in the literature of that among diverse communities, there's almost 0 publications. One way we're getting at this is we're setting the foundation to do a subset of lumbar punctures, so then we can get at some of these pathologies that right now haven't figured out how to get at. But by having the MRI, amyloid, and tau plus the other plasma, we're seeing that mixed pathology or diverse pathology is the rule, right? Us seeing amyloid pathology in the absence of microvascular is rare, very rare. And so I think the rule is mixed pathology. And what I'm hoping is the field will find easier, less invasive ways at getting at alpha-synuclein, TDP-43, et cetera. But I really do believe that that mixed pathology, but even the relative weights of the pathologies themselves are varying too. And so I think that's what we're going to have to get at to better understand in order to advance precision medicine. At least that's my opinion.
I see a question here from Dr. Schnapp, please.
Yeah, we have some questions online, so I'm going to share them. So is the deposition of the increased presence of amyloid in those patients who have it, is it due to increased production or decreased degradation? And can we measure the turnover?
Right. Yeah. And that's a great question. How is it cleared differently among diverse communities? One would think due to different rates of microvascular damage, the clearance probably is different between groups. Those with higher vascular may have lower clearance. With a PET scan, you don't know. With a PET scan, all I know is amyloid. I can't look at, different species, et cetera. So we're trying to get at those sorts of things with the subset of lumbar punctures, but we haven't really done that yet. Our focus groups, for those of you who have tried doing lumbar punctures with underserved communities, you have basically a 90 to 100% no rate. And we did focus groups. We had a 10% agreement rate. And what we found out afterwards is all 10% were interviewed by me personally. And every one of them were like, well, that was Dr. O'Bryant. We said, yes, because it was him, but no, we're not doing that. And so, we're still trying to figure out how to get at that. But it's a question that I can't answer, but it's incredibly important.
Then another question. So it sounds like we've all been taught that race is now a social, not a biological concept. But it sounds like you're seeing biological differences in race. So is race an association, or is it due to other socioeconomic factors?
Yes.
Yes.
It's far more complicated than I think people realize. So we know that just the prevalence of these SNPs varies by race and ethnicity, and that is the presence of the genes or biological construct. I mean, that's not. What does that mean, though? Is what we now see is that the expression of the genes and how they turn into proteins and other factors are absolutely mediated and moderated by environmental factors, because that's what methylation is. Methylation is absolutely heavily impacted by environmental factors. And so it's a mix. It's a mix of biological things. But the thing that I've been most frustrated with from the scientific community is people have been studying the biology of the disease in the absence of the sociocultural factors. And I think what our data is now saying is that the sociocultural, environmental, and that could be, we have data that perceived racism is absolutely related to, for example, neurodegeneration. And so you can't just disentangle and say, well, this is what it is. And so it's a far more complicated system and construct than I think people realized before. And by ignoring, and to me, that's what has led to ignoring race and ethnicity as a construct, was because many people for the longest time felt that race and ethnicity were constructs, were soft science, or the sociocultural was a soft science approach. And that's just wrong. It's wrong. And these factors are of tremendous importance. I don't like getting into the debate of race as a biological construct. I don't think you can disentangle biology from the environmental and sociocultural. They're too interrelated and they're all important.
Thank you. Thank you, Sid. Thanks for making such an outstanding presentation. I echo everything Sterling said. Your leadership and your data are just so unique and so critical for the field. Now, one of the things you pointed out, and I completely agree with you, that the sociocultural factors play a major role. We know that those factors start very early in life. So the significance of life course study of all these factors that eventually lead to the clinical disease. And if that is the case, then these factors act more upstream than downstream in Alzheimer's pathology. And if that is true, we should be really talking about health policy changes, improving education, poverty, environment, rather than discovering anti-amyloid and anti-tau drugs. Just your comments.
You had to throw in that last statement. I'm gonna leave that one alone. I'm gonna let someone else take that one. However, I mean, it's such a good point. The life course nature, I think there was this general assumption that brain aging started at 65. And I don't know, kind of how or where that came from, but obviously to me, when I get asked, when does aging begin? My answer is always at gestation. That's when aging begins. And that's the life course pathway we're on. And so maybe that's a little younger than we want to measure, but that's why we went to 30. And that's why we're capturing residential address over the full life course, because even 30 to me is too late. I mean, personally, I wanted to move the study all the way down to like 5-year-olds, but my team said they would revolt. But absolutely, it's a life course disease. And what we've learned from the heart disease space is the best way to treat a stroke or heart attack is prevent it, right? And we know that early life exposures have a tremendous input or impact on late life heart disease. Well, the same thing's holding for brain health. And we have to get at that, because if we're going to get at true prevention measures are not going to be just some clinical interaction with a physician and a patient. It's also going to be a policy issue, right? Same with smoking, same with heart disease and diabetes. These are also policy issues. And that's of tremendous importance. And studying it from that standpoint, I think, is how we will get at who best needs what intervention at what time in their life.
Thank you, Sid. That was great. A nitpicky question, and then a big picture question that kind of follows up on Sanjay's. The two plasma proteome slides, two different outcomes, the cognitive impairment first and then the dementia second. The nitpicky was that from the plasma collection standpoint, was that cross-sectional or was that longitudinal?
The ones that I showed
Yeah.
so far cross-sectional,
Okay, yeah.
longitudinal data
Yeah,
actually will
'cause
be
that'd
available
really
soon.
important.
And the longitudinal is going to be more important analyses, absolutely.
Yeah, absolutely. The big picture question, or maybe it's a comment, I guess, it's almost a matter of which inflammatory disease is your body exposed to first, earliest in life.
Yeah.
And maybe that's a socioeconomic factor. I was struck by your top 10 in the Mexican American list. Many of them are asthma related.
Yeah, absolutely.
Yep.
Absolutely. And we know that things like asthma are disproportionately impacting COVID, et cetera. And so they all have important impacts.
Wonderful. Can you hear me? Yes, wonderful. That was terrific. Thank you all. One final question just to ask. When you went to your dean and you said you were planning to start this study and you were coming to ask if they would fund it to recruit 1,500 African Americans and 1,500 Mexican Americans, what did your dean say to you? Have you lost your mind?
I've been being told that for a very long time. Sterling pointed out that, I've been doing the blood-based biomarkers. I was told that I was wasting my career at that point. For the health disparities component, yeah, that's what I was told. You can't recruit 1,500 African Americans or 1,500 Hispanics. And now I just look at them and go,
No, you have. Congratulations.
we have.